Hello students. Students, today we will discuss about Lac Operon. So let's see what is Lac Operon. In the year 1961, two famous microbiologists, that is Francis Jacob and Jacques Monod, they proposed a mechanism of regulation of gene expression in E. coli and called it as Operon model. This Operon is of two types, inducible system and repressible system. Today we will discuss only inducible system, and inducible system is that system which requires an inducer. It requires an inducer. Now what is this inducer? This inducer is a chemical. In the presence of this chemical only, the operon will get switched on. Here it is switched off. But when the chemical substance, which is an inducer, it is present, it will get switched on, it will start working. Otherwise, it remains in switched off condition. Here we can see an operon. This is an operon. Operon is actually a part of a DNA. Now this operon has four types of genes present here. What are these four types of gene? P gene, I gene, O gene, Z gene, Y gene and A gene. Here we can see P. P stands for promoter gene. P stands for promoter gene. Now what is the function of promoter as we have discussed earlier? Promoter gene is that gene where the RNA polymerase will bind. Second is gene I. This I does not means inducer, it means inhibitor. Inhibitor gene. This inhibitor gene will undergo transcription and will produce a repressor messenger RNA. Clear? It will undergo transcription. So here it will undergo transcription and it will produce a messenger RNA. This messenger RNA is called as repressor messenger RNA. It will undergo translation and it will produce a protein, and that protein is known as repressor protein. So it is inhibitor gene. This is gene O. It is known as operator gene. If this operator gene, what happens? If the repressor protein binds with the operator gene, then the transcription of this structural gene will not take place and then the messenger RNA will not be formed. Clear? So this operator gene is present adjacent to our structural gene, and if it is binded with the repressor protein, then what will happen? There will be no transcription of the structural gene. Next is gene Z. This gene Z undergoes transcription and it produces the messenger RNA. This ZYA, all these undergoes transcription and then translation and produces the enzyme. The gene Z will produce the enzyme beta galactosidase. And this enzyme is required for breakdown of lactose into glucose and galactose. Clear? So here we can see the gene Z, it will produce the enzyme beta galactosidase, and this beta galactosidase is required to break down lactose into glucose and galactose. Next is gene Y. It produces the enzyme, or it undergoes transcription and translation and produces the enzyme permease. And this permease is required to make the permeability of the cell membrane. As a result, this beta galactosidase can enter the cell. And the gene A, this gene A, it undergoes translation and it produces the enzyme trans-acetylase. So this is the full form of all these genes and what is their function. Again we will see P for promoter gene where RNA polymerase will bind. I for inhibitor gene or regulator gene which will undergo transcription and translation and will produce a protein called the repressor protein. This is the operator gene. This is the structural gene Z, Y and A. They undergo transcription and translation and produces Z will beta galactosidase, Y permease and A transacetylase. These three enzymes are required for the breakdown of lactose from glucose to galactose. Okay? Lactose, it will be converted into glucose and galactose only when the enzyme beta galactosidase is present. And permeases, it causes the permeability, and transacetylase, it is required to transfer the acetyl group from coenzyme A to the beta galactoside. Now here we can see the first condition when the inducer is not present in the medium. First of all, what is inducer? Inducer is a chemical substance. So here, suppose this is the bacterial cell and it is present, and when the lactose is absent, when there is no lactose present, in the absence of inducer, when lactose is absent. Now what will happen when lactose is absent? This gene I, it will undergo transcription and it will produce the repressor messenger RNA. This repressor messenger RNA will undergo translation and will produce a protein called repressor, and this repressor protein will bind to the operator gene. Clear? Now when it will bind to the operator gene, then what will happen? Our RNA polymerase, suppose this is our RNA polymerase. Clear? What is this? This is RNA polymerase. This RNA polymerase will not able to slide over the structural gene. And if this RNA polymerase will not able to slide over the structural gene, what will happen? This structural gene will not undergo transcription and translation, and they will not be able to synthesize these three enzymes, that is beta galactosidase, permease and transacetylase. Clear? And it is not required. These enzymes are not required because lactose is absent here. Clear? So this is the condition when there is absence of inducer. Now second condition we will see when what happens? The inducer is present. Now when inducer is present, that is lactose is present. Clear? Now what will happen when lactose is present? This lactose will be converted. It will enter the cell and it is converted into allolactose. Here what will happen? This allolactose will bind with the repressor protein. Clear? This allolactose, lactose, it will enter the cell. It will be converted into allolactose. And this allolactose will bind with the repressor protein and will form a complex called inactive repressor. Clear? Now this repressor protein has more affinity to allolactose as compared to the operator gene. So it will not bind to the operator gene, it will bind to the allolactose and will form a complex, and it is known as inactive repressor, or we can say it as inducer repressor complex. Clear? Now what will happen? This RNA polymerase which is present, which is binded to the promoter gene, now it can slide over the structural gene. When it will slide over the structural gene, what will happen? The transcription process will take place. Lac messenger RNA will be formed. The messenger RNA which is formed, it is known as Lac messenger RNA. This Lac messenger RNA will undergo translation and it will produce the enzyme beta galactosidase, permease and transacetylase. Clear? And what will happen? There will be the breakdown of lactose into glucose and galactose. This process will continue until all the lactose is used up. And when it is used up, what will happen? Again, this repressor protein, it will go and it will bind to the operator gene, and again it will become switched off. So it became switched on only in the presence of inducer. It will remain switched on until and unless all the lactose is being used up, and after that again the repressor protein, it will go and it will bind to the operator gene, and now again it will turn switched off. So again we will see what is an operon. Operon is the part of the DNA. Now it has 4 types of genes present here: P promoter gene, I inhibitor gene, O operator gene, and the structural gene which has Z, Y and A gene. P gene where the RNA polymerase will bind, I gene which will produce the repressor protein, O is operator gene, Z gene it will produce beta galactosidase, Y gene it will produce permeases, and A it is required for the formation of trans-acetylase. When there is no inducer present, what will happen? The repressor protein will bind to the operator gene. But when the inducer, that is lactose, is present, what will happen? This lactose will enter into the cell. It will be converted into allolactose, and what will happen? This repressor protein has more affinity to allolactose, and it will bind to the allolactose repressor protein, and a complex will be formed that is known as inducer repressor complex. As a result, now this RNA polymerase will be able to slide over the structural gene, which will lead to the transcription. There will be the formation of Lac messenger RNA, and under translation what will happen? It will result in the formation of the enzymes which are required for the breakdown of lactose into glucose and galactose. I think this topic is clear. Thank you.